A very lovely good evening to all of you. Well, this is Dr. Pooja, your original guru of biochemistry. So today we are going to start up with our biochemistry classes. Now, we all know that if we talk about the importance of biochemistry, it is a very important subject. We all know that. And if we see the amount of questions which have been asked in this subject is sometimes it is like 7 to 8, sometimes it is like 2, sometimes it is like 10 also. Okay, so first of all, before starting up with the subject, how we have to go about it? We all know that in biochemistry, what are the topics which we study? The most important topic or the first and the foremost topic which we have to begin with are the enzymes. Alright, after enzymes comes the carbs, carbohydrates, their chemistry and metabolism. Then after this, proteins, again same thing, their chemistry and metabolism, and then the lipids or the fats, their chemistry and metabolism. So this comprises of the first segment of biochem. Second segment includes vitamins, then minerals, obviously their chemistry and metabolism, and molecular biology. So we would be focusing on these important topics or the core of biochemistry in these classes. And how you have to prepare is, again, if we talk about enzymes, the best way to remember biochemistry is you have to remember biochemistry with the help of the mnemonics and the charts, and that is what we will be focusing on, because the charts, when you are going for your exam, if you have your handy charts with you, so what you can do is you can just revise them. And obviously if we talk about the mnemonics is as per your convenience. So these are the two factors which you people should keep in mind to prepare biochem. So today we are going to start up our class with enzymes. Now, first of all, what is the significance of these enzymes? What is the importance, biomedical importance, or what is the diagnostic importance of enzymes? Because if we talk about the questions have been mainly asked on the diagnostically important enzymes. First thing, very important. Then the rate limiting enzymes, the rate committed enzymes, and obviously about different type of inhibition, as you people have seen the pattern of the questions. Okay, so let's begin with our first topic, enzymes. Now, what are enzymes? They are simply defined as the biocatalysts which increases the rate of a biochemical reaction. Now the question is how they increase the rate of a biochemical reaction. They increase by lowering down the activation energy. Very important. So this becomes one question. How enzymes increases the rate of a biochemical reaction? They always increases by lowering down the activation energy. And what is activation energy? We know. It is clear from the word itself. The energy required to activate the reactants or the substrates so that they can be converted into products. And if we talk about the enzyme catalyzed reaction, it is enzyme plus the substrate which forms enzyme substrate complex which dissociates to form the enzyme and the product. Now the question is where the substrate binds to the enzyme. The substrate always binds to the active site of the enzyme. Say this is the enzyme and say this is the substrate. So this becomes the active site. So how do we define an active site? Where the enzyme binds, where the substrate binds or the enzyme takes it. And what happens is it is going to form a enzyme substrate complex like this. It is a complex which is dissociated to form the enzyme back and the product. Then the next important point which we have to remember is that all enzymes are proteins. Very important. all enzymes are proteins except one. So all enzymes are proteins except one, which are the ribozymes. So except ribozyme, as it is clear from the word itself, ribo means where RNA has a catalytic activity. So if we talk about the example of ribozymes, there are innumerous examples. For example, peptidyl transferase, which is a very important enzyme required for translation. Then second important example of ribozyme itself is ribonuclease P. If we talk about snRNAs, they all are ribozymes. So always remember, all enzymes are proteins except ribozymes, and the examples are these. Then third important point about enzymes is that all enzymes require the help of certain molecules. They cannot work on their own, right? So if an enzyme, this is a very important component or the topic from which so many questions have been asked. So this enzyme, when it reacts with a mineral ion, or you can say simple an ion, right, then it is known as a cofactor. Mineral, or you can say metal ion. Metal ion would be the most appropriate one. So if they combine with a metal ion, it is known as a cofactor. And when this enzyme combines with the organic molecule, it is known as a coenzyme. Very important, right? Whenever an enzyme is combining with a metal ion, we call it as a cofactor. Whenever an enzyme is combining with an organic molecule, we call it as a coenzyme. And just the enzyme part is known as a apoenzyme. And this apoenzyme, when it combines with either a cofactor or a coenzyme, is known as holoenzyme. So this is a very important part which we have to remember. So if I'll just highlight it, whenever an enzyme is combining with a metal ion, it will be known as a cofactor. Whenever it is combining with the organic molecule, it will be known as a coenzyme. Okay, and just the protein part is known as a apoenzyme, and when it combines with this, it is known as a holoenzyme. Now the question is, what are the different examples of cofactors and what are the different examples of coenzymes? So first of all, if we talk about the cofactors, the most important point, or on which the questions have been asked, you have to remember it, that copper is a cofactor for cytochrome oxidase. Similarly, iron is a cofactor for cytochrome oxidase. If we talk about zinc, zinc is a very important cofactor for, you can say, alcohol dehydrogenase, generally dehydrogenases. So I'll just write dehydrogenases or alcohol dehydrogenase. Similarly, magnesium or manganese is a cofactor for all kinases except pyruvate kinase. That's an exception. So I'll just write it, potassium is required for pyruvate kinase. So these are the examples of the cofactors, right? Copper cytochrome oxidase, iron के लिए भी cytochrome oxidase, zinc के लिए you can say alcohol dehydrogenase is the example, magnesium के लिए kinases is there, exception is pyruvate kinase. So cofactor is just a metal ion. Then coming over to the next segment, the examples of coenzymes. Now this is a very important part on which generally the questions are asked. So if we talk about organic molecules mean the compounds which have carbon in them. Okay, carbon in them. So most of the vitamins, they function as the coenzymes. Now to begin with, if we talk about B1, the active form of B1 is TPP, which we are going to discuss again in detail under the topic vitamins. Now if we talk about the example of this, B1 is a coenzyme for oxidative decarboxylation, which is a very, very important process which is occurring in the body. Oxidative decarboxylation, which means simply removal of carbon dioxide in the presence of oxygen, as it is clear from the word itself, oxidative decarboxylation. Then if we talk about B2, which is riboflavin, and the active form of B2 is FAD or FMN. So I'll just write FAD or FMN, and they are the coenzymes for electron transfer reactions, or you can say majority of the dehydrogenases reactions. Then we come over to B6. B6, which is known as PLP, is the most important coenzyme for transamination. Very important. Right? Always keep this thing in mind, and this is a question which has been asked in the exam also, that for transamination, what is the name of the coenzyme? So it is always B6. Similarly, along with transamination, it is required for decarboxylation also. And what is the basic difference between decarboxylation and oxidative decarboxylation? The basic difference is, as it is clear from the word itself, decarboxylation means removal of carbon dioxide, but when this carbon dioxide is removed in the presence of oxygen, it becomes oxidative decarboxylation. So you have to keep this thing in mind whenever the question in the exam is asked like this. The coenzyme for oxidative decarboxylation, it is B1. The coenzyme only for decarboxylation is B6. Then coming over to the next one, B7, which is biotin. Now, जैसे PLP is for decarboxylation, B7 is for carboxylation, माने attachment of carbon dioxide. Very important. So the basic difference is B1 is for oxidative decarboxylation, B6 is for decarboxylation, and B7 is for carboxylation. Then we come over to B9. B9 is nothing but folic acid or folate, and B9 is required for 1-carbon metabolism. This is again a very important concept. So coenzymes are simply the organic molecules. So always remember, an enzyme will combine with a cofactor, which is a metal ion. It will react with a coenzyme, which is an organic molecule. Just the protein part of an enzyme is known as a apoenzyme. But when this apoenzyme combines with a cofactor or a coenzyme, it becomes a holoenzyme. Then another important point about the enzymes is that enzymes are highly specific in action. This is very important. Now that specificity can be of any type. So simply you can say enzymes are highly specific. If it is amylase, it has to act on only starch. If it is alpha amylase, it has to be alpha 1,4. If it is beta amylase, it has to be beta 1,4. So this specificity can be absolute specificity, very important again. It can be stereospecificity, or simply you can say they will be highly specific in action. Then after this point, this is again a very important point which you have to remember, that enzymes are highly specific in action. Whether it is absolute, whether it is stereospecific. Then after this, then we come over to, there are certain terminologies on which the questions have been asked. What do you mean by the enzyme activity? What are the factors which can influence the activity of the enzyme? So these are some of the nick and knacks which are generally asked in the exam. So first of all, what is an enzyme activity? So we can simply say that enzyme activity is how active an enzyme is. That how many micromoles of a substrate can be converted into the product in a given time period. So it can be micromoles per liter, micromoles per second, like that. So this is enzyme activity, which is also known as, in general we can say that if the enzyme is more active, its turnover number will be more. So we can simply say that it will be the Kcat, or it is the catalytic constant. So this is again very important. Then after enzyme activity, what are the factors which are affecting the enzyme activity? Factors affecting enzyme activity. So there are basically three factors which can affect the activity of the enzyme. And what are those factors? The most important one is temperature. Then comes pH. And the third and the very important one is substrate concentration. So these are the three factors which can actually affect the activity of the enzyme. Second and third. Now let's consider these factors one by one, because the question which can be asked on this particular topic is that graph which is obtained after seeing the results. So if we talk about the temperature, always remember one thing, that all enzymes work at a temperature optima. Now what do you mean by temperature optima? Temperature optima is that temperature, that particular range of temperature when the activity of the enzyme would be maximum. Okay, so we can simply say that all the enzymes work at a temperature optima, which is actually 35 degrees to 40 degrees centigrade. And whenever we make a graph also like this, so if we talk about the enzyme activity here and if we talk about the temperature here, it will show a temperature optima between 35 to 40 degrees, means here between this 35, 36, 37, 38, 39 and 40. And what kind of graph is obtained? This is a bell-shaped graph. So all enzymes will show a maximum activity या temperature optima between 35 degrees to 40 degrees centigrade. A sudden rise in temperature, because enzyme क्या है? Enzyme is nothing but a protein. So even if we increase the temperature, even if we decrease the temperature, enzyme will denature. Okay. So any increase or decrease will denature the enzyme. This is very important. Then second factor is pH. We all know pH is negative logarithm of hydrogen ion concentration, and pH plays a very, very important role in the body, in a sense that pH is the one which is regulating the acid-base balance. Acidosis is there, alkalosis is there, metabolic acidosis, respiratory acidosis, so many things are there. So pH plays a very important role. Now again, if we talk about the temperature optima, same is the case with the pH. So all enzymes will have a pH optima also. pH optima. Now the question is, what will be the range of the pH? So it is 5 to 9, but there are certain exceptions to it. Okay, so in general, majority of the enzymes will show a maximum activity in the pH optima, in the pH range between 5 to 9. But there are certain exceptions. For example, if we talk about pepsin, we all know pepsin released by the oxyntic cells of the stomach. It works at a very high acidic pH. And what is that pH? Pepsin works at pH 2, which is very acidic, which does not lie in the range. But still, pepsin is a very important enzyme, pepsinogen, which actually starts the degradation of the proteins. Similarly, if we talk about alkaline phosphatase, we all know alkaline phosphatase is a very, very important enzyme, which is a marker also of obstructive jaundice. You know at what pH this alkaline phosphatase works? It works at 11. It starts from 10.6. It's a very high alkaline pH. Okay. So these are the two exceptions, that pepsin works at 2 pH, alkaline phosphatase works at 11 pH, but otherwise majority of the enzymes will show a pH optima between 5 to 9. And if we talk about the graph, it will be similar to what temperature has, 5 to 9, like this. 6, 7, 8 and 9. This is again here enzyme activity, and here it is pH. So the graph is again bell shaped. Okay, then after first, second, then comes the third factor. And if we talk about the third factor, the third is the most important factor, which is substrate concentration. Now, the question is, what is the importance of this? Now, if we talk about any enzyme catalyzed reaction, it is like this. Enzyme combines with the substrate, forming enzyme substrate complex, which dissociates to form the enzyme and the product. So, actually we are concentrating on only the substrate, because enzyme is highly specific about the substrate, and substrate is going to bind only at the active site of the enzyme. Okay, so everything, basically we have to calculate this enzyme substrate complex. If we come to know that how much enzyme substrate complex is produced, then only we are able to visualize the amount of product form, as simple as that. Okay, now the question is, what will be the graph? Just now I told you enzyme is taking the substrate on its active site. So if the enzyme has 10 active sites, only 10 substrate molecules will bind. So if we have to plot a graph, here the substrate concentration is there and here the enzyme activity is there. So if I say that more the active site, more will be the substrate concentration, more will be the rate of a reaction. But a stage comes when all the active sites of the enzyme are occupied by the substrate, then it attains a steady state kinetics. So you can say that more the active site, more is the reaction, right? But when the active sites are completely occupied by the substrate, the enzyme says no to accept more of the substrate. So then it attains a saturation kinetics. This is again very, very important because it attains a saturation state. And it is also known as steady state kinetics. And it is also known as zero order kinetics. So it is basically known by three names कि when all the active site of the enzyme are occupied by the substrate, it is going to give a saturation kinetics because it is now saturated. It is going to give a steady state kinetics because it is steady state now, or it is also known as zero order. Now why it is known as zero order? Because now the enzyme and the substrate are independent of one another. And these three terminologies gave the kinetics of the enzyme, which was actually visualized by two scientists, Michaelis and Menton, which gave an equation, which is again very, very important. You are not going to derive the equation, but yes, the questions have been asked on what is Michaelis-Menton equation. So we will be just going forward to just see the equation, what is it. Okay, so first of all, you have to understand this concept. What is saturation? These are actually the synonyms only. Saturation, steady state, zero order. And what kind of graph is this? If you look at the graph, you can see there is a difference. This is a bell-shaped graph and this is a hyperbola. So hyperbola is obtained whenever we study the influence of the substrate concentration on the activity of the enzyme. So these are the three very, very important factors which are going to influence. Then after this, then we come over to the next important topic here. What is enzyme kinetics? Kinetics we all know. Whenever the reaction starts, there has to be an initial velocity, and when the reaction is accomplished, it forms the maximum velocity. So what is the difference between V naught? So always we say V naught is initial velocity. We designate initial velocity by V naught, and V max, max means maximum velocity. Means when the reaction has just accomplished. Okay, and the reaction is this. E plus S forms ES, which forms E plus P, where K1 is rate of formation of ES, K minus 1 is rate of dissociation of ES back to that, and K2 is rate of formation of the product. And the equation is V naught is equal to V max S over Km plus S. So this becomes Michaelis-Menton equation. V naught is equal to V max S over Km plus S. And the most important point here is if initial velocity is half the maximum velocity, just keep this thing in mind. If initial velocity is half the maximum velocity, you put it in this equation. You can do it in your home also, and you will come to know that Km is equivalent to the substrate concentration. Very important. When initial velocity is half the maximum velocity, Km has to be equivalent to substrate concentration. And if we see in this picture, roughly here, Km will be equivalent to the substrate concentration. So this is initial velocity, or you can say Km is equivalent to the substrate concentration. Now the question is, we are doing so much, we are calculating it, what is the significance of Km? Isn't it? So if we talk about the significance of Km, it is inversely proportional to the affinity of the enzyme with the substrate. So, Km is inversely proportional to the affinity of the enzyme with the substrate. Inversely है, इसका मतलब I can say that if there is any enzyme which has high Km, तो automatically there will be low affinity, and if there is low Km, there will be high affinity. And if you people remember the example, because again this is a very important question which has been asked, high Km, the example of high Km is glucokinase. And if we talk about low Km, the example is hexokinase. Both are the enzymes of glycolysis, but the only difference is hexokinase is always present in the liver. Hexokinase is present in all the tissues, glucokinase is present in the liver. Insulin will always act on glucokinase, not on hexokinase. No doubt we will be discussing the differences in glycolysis in detail, but here you have to know that glucokinase is having a very high Km, it has a very low affinity. On the other hand, hexokinase has a very low Km, it has a very high affinity. So, Km here is inversely proportional to the affinity of the enzyme and the substrate. Okay. Then there was another scientist who gave the concept of reciprocal plot. He basically wanted to differentiate, he wanted to convert it into an equation of straight line, which is known as Lineweaver-Burki's plot. Right. So, this Lineweaver-Burki's plot, it is actually the reciprocal of Michaelis-Menten equation. Of Michaelis-Menten equation. We know the equation. So, if we say V naught is equal to Vmax S over Km plus S, we simply reciprocate it. So, it will come out to be 1 upon V naught Km over Vmax S, you can do it in your home also, plus 1 upon Vmax. Right, and it comes out to be obviously a straight line. So, this becomes Lineweaver-Burki's plot, like this, where this is 1 upon V naught and this is 1 upon substrate concentration. This becomes the intercept and this becomes the slope. So, if we talk about the intercept, this is 1 upon Vmax and this becomes Km over Vmax. So, this is Lineweaver-Burki's plot, which is a reciprocal of Michaelis-Menten equation. Again, a very important question which can be asked in the exam. Right. Then after discussing this, then we come over to another very important topic in enzymes, classification or enzyme classification. Now, as per older system of classification, because I have seen most of the students have asked this question to me many times, that you must have seen in the books that it is written as 1 dot like this, 1:2:2 like this. This was an older system of classifying enzymes, ki jaise this is the class, this is the subclass, this is the group transferred, or to which moiety it is transferred. But this somehow failed. And as per International Union of Biochemical Nomenclature, we classify enzymes into 6 main classes. Now, one more class has been added. So, there are 7 major classes. What are these? Simply, you have to learn this mnemonic as O, T, H, L, TYL. This is the mnemonic, or other mnemonic is our teacher has lied in the class, in the lecture. So it's up to you. Again, it's starting say I'm telling you, you can make mnemonics as per your convenience also. And wherever I'm discussing the mnemonics, if you find my mnemonics good, so you can just remember my mnemonics. Okay. So it is OTHLIL and the last one is T, translocase. So oxidoreductases, transferases, H for hydrolases, L for lyases, I for isomerases, L for ligases, and T for translocases or translocase. Translocase. So these are the 7 major classes of enzymes as per International Union of Biochemical Nomenclature. And this oxidoreductases is considered to be the most elaborative class as it has 4 subclasses. D, O, O, P, where D stands for dehydrogenases, O stands for oxidases, this O stands for oxygenases, and this P stands for peroxidases. Okay, so this is the entire classification of enzymes. Now the question is, what type of questions are asked on this classification? The question would be like that the enzyme would be given to you and you have to find out to which class it belongs. Okay, so this is how you have to remember this, and I'll just try to simplify as much as I can. So if I talk about this 1, 2, 3, 4, 5, 6 and 7. Now first class is oxidoreductases. Now if we see by the word itself, it is self-explanatory, means the enzyme which is catalyzing oxidation and reduction, okay, simultaneously. And this is what is the every you can say redox reaction in the body is like that. So how to remember this? Jaise I told you, it has 4 classes, dehydrogenases. So you will come across these examples so many times. If we talk about dehydrogenases, the most common example is you people should know or must be knowing LDH, which stands for lactate dehydrogenase, right? PDH, which stands for pyruvate dehydrogenase. So wherever you will find dehydrogenase as a suffix, aankhein band karke you have to go for oxidoreductases. But most of the students get confused between the two. Oxidoreductases is the name of the class and dehydrogenases is the name of the subclass. Okay, you cannot just blindly go for dehydrogenases as the name of the class. No, it is a subclass, but the class is oxidoreductases, because this question has been asked in your exam. Okay, then after dehydrogenases comes oxidases. Oxidases mein you can simply write the example of xanthine oxidase. Then if we talk about oxygenases, oxygenases mein there are actually simple oxygenases, mane there is addition of 1 oxygen or 2 oxygen. So if there is addition of 1 oxygen, we call it as monooxygenase. And if we add 2 oxygen, we call it as dioxygenase. And if we talk about monooxygenase, you have to simply remember the example as hydroxylase. Wherever, because if you remember, hydroxylase means OH, only 1 oxygen is attached. So example can be phenylalanine hydroxylase. Okay, tyrosine hydroxylase. And if we talk about dioxygenase, the most important example is homogentisate, which you people have studied and you must have seen the question also. Homogentisate oxidase, which is also known as dioxygenase. Right? And the fourth one is simply peroxidase, CDOOP. Peroxidase. And if we talk about peroxidase, peroxide H2O2. So we can simply say that peroxide mein glutathione peroxidase is the name of the enzyme, which again plays a very important role in maintaining the fragility or integrity of the cell membrane of RBC. So glutathione peroxidase. So this is our first class of enzymes, oxidoreductases, which has 4 subclasses: DOOP, dehydrogenases, oxygenases, oxidases and peroxidases. Then comes the second class. Second class is transferases. Okay, transferases. Now, if we talk about transferases, it is clear from the word itself, transfer hona. Isn't it? Transfer of what? So, if there is a transfer of phosphate, we call it as kinases. So, kinases is transfer of phosphate. An example is glucokinase, wherever you will find kinase written. Fructokinase, hexokinase, galactokinase. So, they will come under the class, subclass kinases. Okay. Then, second important example is transfer of an amino group. So, it is aminotransferases. So, if we talk about aminotransferases, the most common example is ALT, which is alanine transaminase, commonly which is known as SGPT, and AST, which is commonly known as SGOT. And the third example here is there is a transfer of phosphate, but it is known as phosphorylatic splitting. And the example of this is phosphorylase, which is glycogen phosphorylase. We all are familiar with this enzyme, but we are going to take this enzyme in detail later in the metabolism of glycogen. So, glycogen phosphorylase. So, this is class second. Then OTH. Then comes H, hydrolases. Hydrolases may we can simply quote the example of all the digestive enzymes. All the digestive enzymes will come under hydrolases class. So, I am simply writing digestive enzymes. And these are chymotrypsinogen is there, pepsinogen is there, whatever amylase is there, lipase is there, nuclease is there. So, depending upon the substrate, they will be classified like that. Then OTHL. So, L is lyases. Now, in general, we will call lyases as a splitting up class. So, commonly we call it as splitting up class. And if we have to take the examples of this, we can simply say that example is the most common example is aldolase. We all know aldolase A and aldolase B. They are the splitting up enzymes. If you remember, aldolase A is the enzyme used in glycolysis and aldolase B is in the metabolism of fructose, which obviously we will be discussing also. Okay, and fumarase. So, these are the 3 important examples which come under lyases or the splitting up class. So, OTHL. Then we come over to I, means isomerases. Now, see, isomerases means which is catalyzing the isomers which are I mean like the enzymes which are actually converting one isomer into another. And if we talk about isomerases, there are so many examples. जैसे, for example, if we talk about mutase. Mutase में the most important example is phosphoglucomutase. This question has been asked in the exam. I'll just write a question mark, phosphoglucomutase. And the reaction is what? Simply conversion of glucose 6-phosphate to fructose 6-phosphate. Mutase, sorry. So it is glucose 6-phosphate to glucose 1-phosphate. Glucose 1-phosphate. Because always remember, mutase mane there is shift of phosphate from first position to sixth position. Okay. Then second is isomerase. Isomerase mein we can quote the example of simple. Example is phosphoglucoisomerase or phosphohexoisomerase. Phosphohexose isomerase, where there is conversion of glucose 6-phosphate to fructose 6-phosphate. And the third example is racemase. And if we talk about racemase, racemase mein the example is conversion of, I'll just write conversion of D-methylmalonyl-CoA to L-methylmalonyl-CoA. So it is D-methylmalonyl-CoA to L-methylmalonyl-CoA. Okay, so I can say isomerases mein there will be three examples: mutase, isomerase and racemase. So it is like OTHLI. Then the second last class is ligases. Now if we talk about ligases, in general we call this ligases as a sealing up class. It seals up the things. Okay, so this is sealing up class or synthesizing something. Sealing up class or synthesizing. And the most common example of ligases is DNA ligase. We all know DNA ligase is known as glue of DNA. And if we talk about synthesis, just now I told you, to synthesis mein aa jayega, the example will be glutamine synthetase. Glutamine synthetase is a very, very important enzyme which obviously we are going to discuss when we will study the metabolism of these amino acids. But here I just want to tell you, as it is clear from the word itself, glutamine synthetase mane synthesis of glutamine. Right? So these will come under ligases. Then comes the last class, translocases, which has been just recently added. Translocases or translocase. ये क्या करता है? It transfers the substrate from one part to another. And the example of translocase is endoplasmic reticulum translocase enzyme. Translocase. And what is the function of this? This simply transfers glucose 6-phosphate to endoplasmic reticulum where glucose 6-phosphatase is there. So basically what happens is this glucose 6-phosphate is transported so that this glucose 6-phosphatase can act and it can be converted into glucose. So this is translocase. Right? So overall, if we talk about as per biochemical nomenclature, there are now 7 classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases and translocases, right? So this is about the classification of the enzymes. Then there are again certain important subtopics which you should know. What is the difference between synthetase and synthase? Okay, so this is also very important because most of the students get confused between the spellings. Difference between synthetase and synthase, right? So if we talk about synthetase, it always involves ATP, always involves ATP. Example is glutamine synthetase, and another example can be carbamoyl phosphate synthetase 1, or you can say carbamoyl phosphate synthetase 2. And if we talk about synthase, never involves ATP, never involves ATP. An example is citrate synthase. Okay, so this is the difference between synthetase and synthase. Then another important point about this is, which you people should know, what is kinase? What is the difference between kinases and phosphatases? There is one more terminology, phosphorylase. Right. So if we talk about, because this is the type of terminology which actually creates confusion. So if we talk about kinases, always remember kinases attaches inorganic phosphate. Always remember. Phosphatases digests or removes phosphate. And phosphorylases transfers phosphate in with the product. जैसे if we talk about conversion of glucose to glucose 6-phosphate, it is clear phosphate is being attached. So here the enzyme is hexokinase, and ATP is converted into ADP like this. So this is kinase. If we talk about phosphatase, glucose 6-phosphate will be converted into glucose. Here the enzyme is glucose 6-phosphatase. Phosphate is given out like this. And if we talk about phosphorylase, phosphorylase converts glycogen to form phosphorylase. It will simply give glucose 1-phosphate. So you can see the difference between the activity of these three enzymes. So glucose is converted to glucose 6-phosphate by hexokinase. Glucose 6-phosphate, when it is removing phosphate, the enzyme is glucose 6-phosphatase, and glycogen, in the presence of glycogen, phosphorylase is going to form a product which is glucose 1-phosphate, and glycogen so produced will have one glucose molecule, a residue less than glycogen. Okay, so this is another very important point. Then after this, then we come over to another segment. What is the difference between rate limiting and rate committed enzymes? because you have to make a chart of the processes on one hand and about the rate limiting or rate committed enzymes on the other hand. Okay. Now it is clear from the word itself, rate limiting. Mane, it is going to limit the rate of a particular reaction for some time, which is none other than your feedback inhibition. But rate committed enzyme is that enzyme like you people are committed to crack the exam. Okay? So for example, rate committed enzyme will be that enzyme ki whenever a substrate has entered and the enzyme has acted upon it, uska fate will be only one reaction, not other reactions. For example, if we talk about this reaction, conversion of glucose to glucose 6-phosphate. This is a reaction of glycolysis, and we can take the third reaction, conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Right. Here the enzyme is hexokinase, we all know, and here the enzyme is PFK1. Now the question is, which is a rate limiting and which is a rate committed enzyme? Now see, if we talk about whenever there is increased concentration of glucose 6-phosphate, it will inhibit hexokinase, so it becomes rate limiting. But if we talk about glucose 6-phosphate, it cannot be rate committed. Why it cannot be rate committed? Because it is attached, it is linked to so many other reactions at the same time. Means glucose 6-phosphate can enter glycolysis, it can enter glycogenesis, or it can enter HMP shunt. Means glucose, whenever it has to enter in the body only, it has to actually undergo so many different paths. On the contrary, if we talk about this, सबसे पहले, if there is increased concentration of fructose 1,6-bisphosphate, obviously it will inhibit its own reaction, which is feedback inhibition or product inhibition. So it is rate limiting. But if we talk about the fate of this, fructose 1,6-bisphosphate has only one fate, that it can enter only glycolysis, not any other reaction. So I can say that this is rate committed and rate limiting both. Limiting, right? Whereas this is only rate limiting. Not rate committed. Very important. Right? So this is the basic difference between rate limiting and rate committed enzymes, which you people should know. So always remember, suppose if I ask you a question, which is the rate limiting enzyme of glycolysis, you can always go for hexokinase, phosphofructokinase, pyruvate kinase. But if the question is asked in the exam like this, that which one of the following is a rate committed enzyme? So there is only one rate committed enzyme of glycolysis, which is phosphofructokinase 1, right? Then after this, then we come over to the next important topic in enzymes, enzyme inhibition. Now, if we talk about enzyme inhibition, it is clear from the word itself, inhibition means an act of stopping, isn't it? What is inhibition? An act of stopping. Now, how it can stop the reaction? Because the enzyme is combining with the substrate, as I have told you, forming enzyme substrate complex which dissociates to form the enzyme and the product. So in what conditions it is stopped, how it is stopped, and what is the significance of this stopping? There is so many therapeutic use of this also. So if we talk about this enzyme inhibition, we can simply classify this into 4 subheadings. Reversible, because there are different ways of classifying. Different classification are given in different books, but we will be only concentrating on the ones which have been asked in the exam. So reversible is there, irreversible is there, suicide inhibition is there, and the 4th one is allosteric. So there are 4 type of inhibition: reversible, irreversible, suicide and allosteric. Now, what are the different examples in this? So if we talk about reversible, it is clear from the word itself, it can be reversed. Okay. So here the example will be competitive inhibition. If we talk about irreversible, the example is non-competitive. Now there is a controversy regarding this non-competitive. It can be reversible, but most commonly it is irreversible. Okay, so if the question is asked in the exam, you always have to go for non-competitive inhibition as irreversible inhibition. Suicide inhibition is different, allosteric is different. Okay, so first of all, we are going to study this competitive inhibition. What is it? And what is the kinetics of the enzyme in it? So, as it is clear from the word itself, there is a competition. Kisme competition hai? Between the inhibitor and the substrate. So, there is actually a fight between the inhibitor and the substrate, that who's going to bind first with the enzyme, right? So, if we talk about this, if we see the equation is enzyme, this already we have discussed, combines with the substrate to form enzyme substrate complex. Now the question is, the inhibitor is coming, and which is resembling the substrate in structure. So what will happen is, if the enzyme combines with the inhibitor, so it is going to form enzyme inhibitor complex, means no product formation is there. Very important. So I can say that since there is a competition between the inhibitor and the substrate, so instead of taking the substrate, it can bind with the inhibitor. Now what will be the kinetics of the reaction? So if we talk about Km, Km will increase. Now why Km will increase? Because the affinity of the enzyme with the substrate will decrease. And if we talk about Vmax, Vmax will remain unchanged, or it will be the same. Why it will be the same? That whether the inhibitor is acting, whether the substrate is acting on the enzyme, the reaction is going to in a normal phase. This is very important. Okay. So competitive inhibition mein there is a competition between the substrate and the inhibitor, where Km always increases and Vmax remains the same. And if we have to plot a graph, so it will be like this. It will be the same. Okay. Like this. And if we talk about the Km, there will be a change. So this is competitive inhibition. And if we talk about the Line-Weaver-Burkes plot, we can simply say that there will be a change in Vmax, in Km, but Vmax will remain the same. So this will be the equation for Line-Weaver's-Burkes plot. And this will be, so So I can simply say that a X-shaped graph will be obtained in competitive inhibition, and this will be the type of graph again obtained. This is Michaelis-Menten equation and this is Lineweaver-Burkiss plot. Rest we can label 1 upon V naught, 1 upon substrate concentration. And now the question is, what you have to remember is this. Km increases whereas Vmax remains the same. Okay. Now the question is, what are the examples here? Now the examples which you have to remember here are, सबसे पहला example is HMG CoA reductase is the name of the enzyme, and if we talk about the inhibitors are statins. Statins. Second is, if we talk about second example, we can take that folate reductase, and the example, the inhibitor will be methotrexate. Right. Then third example can be xanthine oxidase, and the inhibitor is allopurinol. Now this allopurinol comes under two examples. One is competitive inhibition and second it will be quoted under suicide inhibition. Okay, so if we talk about the pharmacological action of allopurinol, it has to be competitive inhibition, but biochemical role is that it is actually known as a drug of choice to cure gout. So it is like suicide inhibitor. Okay, that we will discuss separately also. And if we talk about the fourth one, the enzyme is succinate dehydrogenase, and the inhibitor is malonate. So always remember, whenever we talk about competitive inhibition, just keep one thing in mind, Km will always increase whereas Vmax will remain the same, and this is the therapeutic use of competitive inhibition, where you can see that there is a competition inhibitor and the substrate. Why? Because inhibitor is resembling the substrate in structure. Okay, so Vmax is going to be the same, but yes, since there is a competition, Km will increase. And why it is reversible? Because if we increase the concentration of the substrate, the reaction can be reversed. मतलब substrate इतना बढ़ा देंगे कि there won't be any chance for the inhibitor to bind with the enzyme. Then second is non-competitive inhibition. Now, if we talk about this non-competitive inhibition, there is no competition, as it is clear from the word itself. So what will happen is, it's the wish of the inhibitor where it goes and binds. So this is the reaction. So what is happening is, the inhibitor can go and bind with the enzyme, forming enzyme inhibitor complex, or it can bind with the enzyme substrate complex, forming enzyme substrate inhibitor complex. But generally what happens is, this becomes the scenario, and that is why non-competitive inhibition is known as irreversible inhibition. Because once the inhibitor binds with enzyme substrate complex, the matter is finished. Okay. Now the question is, what will be the kinetics? So if we talk about the Km, because these are the two factors which are influencing the entire kinetics. So if we talk about Km, Km will remain same or unchanged. But yes, if we talk about Vmax, Vmax will decreased. And if we have to plot a graph will be, first of all, I will just draw the Michaelis-Menten equation graph. So it is like this, because here what is happening is Vmax is lowered. This. So, and if we talk about Michaelis, this Line-Weaver-Burkes plot, it will be a V-shaped graph like this. Okay, rest you can label here. Like this. So, a V-shaped graph is obtained in non-competitive inhibition by Line-Weaver-Burkes plot, and here it is. You can simply say that this type of graph is obtained. Then we come over to the examples. Now, if we talk about the examples, the most important example is fluoride. Fluoride ions are the inhibitors of enolase enzyme. Enolase enzyme which converts 2-PGA to PEP. This is a very important question which has been asked in the exam also. And the question is asked like this: since fluoride is an inhibitor of enolase, whenever we collect blood sample for glucose estimation, whenever we estimate, I'll just repeat it again, whenever we estimate blood sample या glucose for diabetes, what happens is we always add an anticoagulant along with sodium fluoride. What is the purpose behind that? We always use grey colored vials. Why? To stop glycolysis. Because whenever the sample is there, it has to be processed, right? We know the life of RBCs is 120 days. So what will happen is we have to take the sample from the house to the primary health center. So in that bargain, again, the reaction will keep on going. The moment we add sodium fluoride and anticoagulant, glycolysis is stopped. And whatever is the correct amount of glucose, that can be estimated. So this is non-competitive inhibition, where simple example is that Km, as you can see from the graph, Km is same but Vmax is lowered, right? Then third is, there is a very rare inhibition जिसको हम बोलते हैं uncompetitive inhibition. Now, uncompetitive inhibition में you have to just remember 2 points, that the inhibitor will always bind to the enzyme substrate complex. जैसे this is the equation, enzyme plus the substrate forming enzyme substrate complex. Here the inhibitor will bind only here. So what happens here is Km and Vmax both are lowered, right? This is uncompetitive inhibition, and there is only one example. If we talk about this, the example is there is inhibition of the placental phenylalanine enzyme. Okay, so example is inhibition of placental phenylalanine hydroxylase. Right, and if we talk about the graph here is like this. This is like this, like this. And if we talk about Line-Weaver-Burkes plot, it is parallel, like this. And rest of the things you can just label. So this is uncompetitive inhibition. So if we have to just clearly differentiate between competitive, uncompetitive and non-competitive, there is a difference in the Km. So what are the differences? This is competitive, non-competitive and uncompetitive. Clear? So the two parameters which we have to take to differentiate them, Km and Vmax. So in non-competitive, Km increases, Vmax is same. In non-competitive inhibition, Km is same, whereas Vmax decreases, and in uncompetitive, both decreases, right? So this is the different type of inhibition. Then after this, then we come over to the next one, which is suicide inhibition. Now because most of the students get confused between competitive inhibition and suicide inhibition. The most important difference is that एक तो suicide inhibition में there are only two examples and the product so formed is more potent than the original one. Okay, I'll just explain it with the help of an example. सबसे पहले I'll just write the examples. There are only two examples of suicide inhibition. One is So far we have finished up with the major portion in the enzymes. Now we are going to take 20 minutes break. After that we will continue with suicide inhibition and allosteric inhibition. So see you guys after 20 minutes.